All right, a couple announcements before I get started. I want to set your expectations a little bit low. I'm just getting over a cold. I don't normally sound like this, so sorry about that, number one. Number two, I realize because this is a Python audience, you might think I'm going to talk about some interesting Python projects at Cloudera, like, for example, you can now take the continuum Python distribution and put it in CDH. I'm not going to talk about interesting Python-related things like that. In fact, I figured since it's PyData, it's about Python and data, and maybe we shouldn't do both at once, first thing in the morning. So in fact, there's going to be almost no Python content in this talk whatsoever. I hope that's OK. Lastly, this is a talk about genomics, and I'm not a genomics expert either. People I work with are. So I can tell you the interesting things about things we're doing with genomics at Cloudera, but please don't ask any clever questions. I probably won't know the answer. Anyway, so with all that level setting accomplished, let's get into it. Let's talk about genomics, and I'm going to argue this is really a data problem at heart. Genomics is, well, the study of our genome. And I hope you're thinking now, what's a genome? Well, I know this, right? I think a lot of us studied this at some point in school or have heard about this, but what is the genome and what is the study of it? Well, quite simply, organisms out there like us, we all have a genome because inside of us we have cells. I think you remember, you're with me so far, I think, and inside the cell we have, at some level, a nucleus with some genetic code in there, DNA, right? And for humans, if we looked at our DNA, we would find we have 23 pairs of strands of genetic code. And they're encoded with these base pairs, A, C, G, and T. They're like strips of data, or code, if you will. And how they're represented with amino acids is maybe not important. For our purposes, what's important for us is what they say or what they express, what the genetic information they encode. And that's really our genome. It's the genetic information that this DNA in our cells encodes. So this exists inside all of us, inside of all our cells, and it's amazing when you think about it. But without losing much, we can really think of our genome, or our genetic code, as one big, long strip of genetic code, of these base pairs. And it's billions of base pairs long, so it's a very long, one-dimensional string made out of 4 characters. So that's interesting. But maybe what's really surprising is how similar our genomes are. Not just to each other, but even to other organisms. It's actually, we're not that different from other forms of life on the planet. And so certainly if you look at the genome from different human beings, you'll find they're very similar, like 99.99% similar, something like that. And what that means is because we're so similar, we can begin to talk about common areas of the genome. Because they look so much alike, we can recognize features in one genome that clearly correspond to features in another. That's interesting, that's powerful, but if you take that a step further, it means that we should be able to establish some kind of reference genome. If we're all so similar, we should be able to find some median or a medoid of all human genomes and call that the reference human genome. And maybe that would be powerful or interesting. So you may remember I think about almost 20 years ago now, this announcement of the completion of the Human Genome Project. Bill Clinton sits up there with Craig Ventnor and announces that we've decoded the Book of Life. And this is really what they were talking about. They managed to, for the first time, get a rough draft of the reference human genome. And it took about a decade, and it cost about $2 billion. But this is the first time we had gotten some look at what we thought was the genome for humanity. And that was actually a big step forward. But because once we can talk about a genome, this common string of data that's within all of us, we can then define all of ourselves in terms of our differences from that common genome. So it was cool that we could even do this and sequence the whole genome, but a good step forward for sure. But what that gave us was only the beginning. I think what we get out of something like the Human Genome Project is something like the equivalent of satellite images. So this is a satellite image from Google Maps, and even though I used to live very close to where this is, I didn't recognize it. because this isn't really how we think about places on the Earth, even though this is literally a picture of a place on the Earth. I don't know if anyone's going to recognize what this is. Even I didn't. Unless maybe we start to label it. Did someone guess? It's Washington Square Park. Okay. Wow, very good. Yeah, that's indeed. This is Washington Square Park in New York City. So it's pretty obvious once you turn on the labels in Google Maps, since it's annotated, right? It's actually called Washington Square Park there. But even if you didn't know that, now we see 8th Street and the West 4th Street Station and so on. And it's these annotations that are really the useful bit of the map. I mean, I'm guessing that you and I don't really use the street maps in satellite mode. We use them in this maps mode. We don't really literally care about what's there. We care about what's logically there. The station, the store, this intersection, this service, and so on. So I think this is, by analogy, the next step we had to take in genomics to make use of it. We had to go from just this raw map of the genome to some logical representation of it. What does it mean? What's there conceptually in the genome? Not literally, but logically. So that is, we wanted to go from this, literally a look at the base pairs in the human genome, to something like this. This is maybe a rough equivalent of Google Maps for the human genome. It's a browser, and I appreciate it's really hard to read, even if you can see it. But what we've got here is, it's like here's the zoomed out view of the human genome at the top. It's a couple billions of base pairs long, and that little highlighted red region is the bit we've expanded here below. It's about 600,000 base pairs wide, this bit we've expanded. And we've zoomed in on this, and you can see annotations for different sections of the genome. So if you zoomed in further and further, you could see, I assume at some level, actual base pairs, but at this level, we see more interesting annotated features. We believe this section of the genome codes this particular attribute or is related to this disease, and so on. And it also, interestingly, has some other funny comparisons with, for example, a rhesus monkey, a mouse, and a dog, showing bits of our genome that we can map to other animals' genome as well. It's similar enough that we can even draw those analogies, which is kind of crazy. We're actually apparently a little bit similar to lampreys in some level. That was shocking to me. Anyway, so this is where we want to go with genomics. We want to create more and more of these labels. We want to extract more and more meaning from the genome by understanding how their genome varies, and then mapping that to observable attributes of people so that we can understand logically what pieces of the genome encode for humans and understand better how we operate, how we're built. So this is where I want to connect it to a data problem, because this is all very interesting, but this is mostly, so far, a bioinformatics problem. It sounds just like biology and genomics and so on. What does this have to do with data? Well, I think actually data and computing have been the key to progress in this field. It's not just that better science, better techniques and so on have made it easier and more effective to sequence the human genome. It's the technology has also driven that cost down as well. This is a chart of the cost to sequence a single human genome. It's on a log scale note from 2001 to about this year, and it's dropped from about $100 million down to about just over $1,000, which is crazy. And here's the Moore's Law trend here, and you can see that it's not just advances in hardware getting faster or maybe breakthroughs in the science, but there's also been some big breakthroughs in applying technology to this problem that have led to these order of magnitude drops in the cost. And when the cost goes down this much, things can qualitatively change. It means we don't just get more genome sequenced for cheaper, we get better results. We understand more. So why is that? Why is this such a data-driven problem? Well, at some level, it's because the genetic code is a big string, and so it is inherently something we can represent as data and maybe manipulate as data. But there's a little more to it than that. I think you'll find that if you look at how these bioinformatics processes work, they're not just data intensive, but compute intensive. So a lot of the process of sequencing a genome involves chopping it up into pieces, getting a lot of little chunks of DNA, and then piecing them back together, as if we'd gotten a map and put it in a blender, and then pasted it back onto a board, onto some kind of reference map of the world. So there's a lot of data moving there, but there's a lot of compute going on as well. And so the more we can apply faster computing and smarter computing and data processing to this problem, the faster it's going to get cheaper and more effective. And maybe the more we can do with it as a result. So really, bioinformatics has, in the last decade, really been an informatics problem. It's a data science problem, and it's a data problem. But we still have a lot to do. I think you won't be surprised to learn that inside these bioinformatics processes are pipelines. I think everyone in the room probably operates a data pipeline of some kind. There's data lands somewhere, and something transforms it, and something else transforms it, and then something else is stored here and computed and put there. Pipelines are everywhere, and certainly that's true in bioinformatics as well. For example, one of the first steps is to take all these reads and align them, put them onto a reference genome. So the details don't even matter that much here, but suffice to say, there's a bunch of steps that have to happen to get from raw DNA to understanding what's there in the genome. So people have built software for this, but maybe it won't surprise you to learn that it's been built by people that aren't big data people. It's been built by researchers. It's been built, therefore, ad hoc across many years, across many different research groups that are maybe competing or maybe just don't know each other exist, that know different languages, that have different ideas about what's important, and so on. So I think the state of genomics software technology, such as it exists or has existed maybe a couple of years ago, was your typical mess. You had, for this pipeline, you had a bunch of different tools, a lot of times overlap amongst tools, different groups building different tools for the same activity. You had, it seems, more formats than even intermediate stages somehow. So everyone defines their own file format to encode the inputs and outputs of the different stages, making it really hard to even share one tool or plug one tool into the other. And of course, everyone writes in their own language. Sometimes people write in Java. Sometimes people write the compute-intensive phases in some kind of specialized HPC system. Sometimes some native code. Sometimes all of the above. It's just how it is, but obviously we could do better. I mean, after all, it's not like pipelines are anything new to the big data world. Every day we manage flows of information that consume one type of data and output another type of data. And it's not as if we can't homogenize these things and make these things standard and standardize on formats, but we just haven't done that yet, not in genomics. So just to throw a couple more stones, this is an example of the VCF file format. I even forget what this encodes, since I'm not an expert, but I think you can get a flavor for why the data formats themselves might even be a problem in this field, or have been a problem in the past. So for example, the file format such as it is I think a gzip compressed file, which isn't splittable. I mean, you can't break up a gzip file and process pieces of it independently like you could a text file or a bzip file. The format looks inspired a little bit by mainframe files. You've got a header section and then records, so I'd call it semi-structured. The header records themselves have some structure that differs from field to field. In some cases, this format is underspecified, so it's not clear what you can and can't do with the format. In some cases, it might be overspecified. For example, I think these are maybe reads, and they are supposed to be defined in sorted order. So when you output this file, you have to put it out in sorted order, even if that's expensive to do and maybe not necessary to some downstream consumers. So I don't mean to criticize so much whoever came up with these formats. I'm sure there were reasons. But I think everyone in the room who's dealt with data would look at this and say, this isn't how you would design a representation of whatever this is. today. So maybe we can do better, and maybe it's worth doing better. Likewise, if you look at some of the source code for these projects, this might be from an early version of GATK in Java. And you dig into some of the low-level code here, you'll find a lot of mixing of concerns. For example, this high-level library has a parameter to control the number of file handles that get opened on the host operating system. And I'm sure that there's a reason for that. I'm sure it mattered for performance at some level or at some time. But it just doesn't seem like something that application-level authors should be worried about. Excuse me. This is something we're accustomed to libraries taking care of for us. It's not clear that someone who's writing genomic software would even know how to set this. So okay, so we've got some legacy code on our hands. We've got legacy formats. What can we do? How can we do better here? Well, before we get to solutions, let's even expand the problem a bit. This is a pipeline for one person. This is what we would do to get from one person's raw sequencing data to some annotation of the variants in their genome. We don't want to do that for just one person. We'd like to do that for a lot of people, everyone in the room, maybe everyone in a whole country, for example. So sure, we can parallelize this. I mean, we could run a process for your genome on one machine and your genome on another machine. But that's not necessarily that efficient. For example, some of these stages are very resource intensive and some are not. Some are I/O intensive, some are not CPU bound. And it's probably not that efficient to try and cram each of them onto one machine. We can do it, but it's kind of the best we can do given this current model, given this ad hoc pieced together pipeline for genomics that we've got on our hands at the moment. But for example, in the UK, there's a project to sequence 100,000 genomes. Doing the math, given the current cost, that's hundreds of millions of dollars to sequence 100,000 people's genomes. So it seems like it would be worth it to see if we can do better and do this more efficiently. Because if we can't do 100,000 people, we're not going to be able to get to 1 million or 10 million. So this is why I think people have, in genomics, have started to take seriously big data tools. Because in a way, there's this whole other audience that's been building out infrastructure for large-scale, cost-effective, distributed computation for years. And it's gotten quite good. So maybe we can put the two together. Sure. So, for example, it's not just that parallelizing some of these individual stages might be advantageous. It may be that by combining the results of many people's pipelines, we can produce a better result than any of them individually. For example, it turns out that variant calling works a lot better if you can see everyone's genome data and not just process them individually. And likewise, the recalibration step also works better if you can use calibration data from all the genomes at once and not just doing them individually. Of course, this adds a lot more complexity. And this is beginning to look like a directed acyclic graph of jobs that need to be executed across a bunch of nodes with the shuffles and so on. And I think we've heard that story before. Before we get into computations, we might even start by asking, why can't we use more modern data formats? For example, why make up serialization formats ad hoc? Why not leverage formats like meta-formats like Avro and Parquet that let us define well-structured formats and then let them deal with compression, let them deal with the representation, and so on? So for example, rather than write a bunch of code to represent the VCF format, we could just write a very similar record definition in Avro, for example, or Parquet, and have it generate all the code and manage all of the file formats for us. So this has been done for common formats like BAM and VCF, and you can see that just letting a specialist format deal with compression can save a bunch of space out of the box. Likewise, we've already got answers for distributed computation and distributed storage, Yarn and HDFS, and now we have Spark as well to structure these computations. It's a perfect fit for these types of jobs that need to shuffle a lot of data, need to use results from one stage and another stage, and so on. So maybe it's no surprise that people have started to build on top of this stack that has been evolving outside of bioinformatics for years with Hadoop and HDFS and Yarn and Spark, build on top of that a new generation of tools that use all that infrastructure and just focus on the genomics problems. So maybe it won't surprise you to learn that there are already open source projects for this. One of the big ones is Atom. It's really an implementation of a lot of the pipeline I've mentioned to you, but in terms of standard big data technologies, like Spark especially. So there's a little bit of Python in here, I think, as well, so maybe it's sort of relevant. If you're interested, go look it up. It's from AmpLab, the same organization that brought you Spark in the first place. It's an open source project that anyone can jump in and contribute to. So the nice thing is once we have all of the infrastructure taken care of for us with the right formats, with frameworks like Spark and so on, the projects and the code get a lot simpler. They can just focus on the higher level logic. So this is, a lot's going on here. This is what would have been miles of code before in these other projects. Now it's a pretty simple Spark job to do some of the alignment and deduplication. So this is something you can even access in an interactive shell because it's Spark-based as well. So I think it's a big win, bringing the standard big data tools to the world of genomics. And that should not just drive the cost down and make it more efficient to run these sequencings, but also let us do more complicated things, maybe push the boundaries of what we can, the information we can glean from this genomics data. For example, this is something I'm told is hard to do in general, or has been hard to do in the pipelines earlier, a spatial join, where you actually try and bring together bits of genetic information based on where they are in the genome. This gets quite trivial in something like Spark that can easily shuffle and join data efficiently at scale. So that's an example of how we can not just do things faster, but do things better by leveraging some kind of modern framework. And again, because this is based in Spark, you Python users out there can also access it through PySpark. Just a quick set of benchmarks to show that, hey, this actually does scale pretty well. If you throw 4 times more machines at it, you can, roughly speaking, sequence 4 times as many genomes in the same amount of time. So this scales pretty well. That's good because Craig Ventner's new goal is to sequence 1 million people's genomes by 2020. We're only about 10% of the way there. To get there in a cost-effective way, we are going to have to get better at this. So I'm glad these efforts are underway. And before I close, I do want to give credit where credit's due. If you are interested in these topics, if you think, hey, I'm a Python developer, I'm a Spark developer, I'm not a genomics person, but I'm interested and I want to help out, you're probably the kind of person these projects need most. These are being driven by my colleagues Uri at Cloudera, a lot of folks from UC Berkeley working on the Atom project. I think if you walk up to these projects and look for ways to contribute, you'll find you can. These projects need more data engineers than biologists at this stage. So thank you to these folks for their work and for the presentation. Thank you. I want to keep it short. That's all from me. I think we might have a couple minutes for questions. As I said, no clever questions about the biology, but if it's about Spark, I might be able to answer it. Thank you. Thank you.
One short question, though. Like for long-term observation, I mean, if you use Afro or something like that, will that still work in 20 years? Are there any concerns in that field? Or?
So the question is, I think, format rot. We want this data to be accessible not just next year, but 20 years or 200 years down the line. Is Avro a good bet for that? It's probably a better bet than some ad hoc made-up format like VCF or BAM. So in that sense, yeah, it's probably better to store in some kind of more standard format than not. But I think that's a really good question. Are these formats going to be readable in 20 years? I think the nice thing is formats like Avro are pretty well-defined and self-explanatory. They don't require cryptic custom code to decode. Certainly, and they're open source and they're openly defined. So probably our best bet. Great. Well, thank you very much. Welcome to PyData.